Welcome to Botany 101 Exam Review for Exam 3. I'm Angela Nishimoto, okay? And today's review is going to be covered in two parts, right? We're going to have a part 1 and a part 2. Right now we're going to do part 1, okay? So this is Botany 101 Exam 3 Review, okay? So first of all, one of the things we're going to do is we're going to cover the organisms that are studied by botanists, including bacteria and archaea a little bit, okay? And then we're going to go into the protista, and then we're going to go into the fungi, then into the plant kingdom, okay? So all of these kind of organisms are studied by botanists, right? Now, features of bacteria and archaea include that they are prokaryotic cells. What does that mean? That means pro is before and karyon means nucleus or kernel. So these are prokaryotic cells that have no nuclei, no nuclear envelope, okay? So a couple of images here, one of a flagellated bacterium, a drawing, and the other looks like a micrograph of bacterial cells. Okay, so they have no nuclear envelope. Their genetic material is in direct contact with the cytoplasm. Okay, there are also no membrane-bound organelles in these organisms. Okay, furthermore, they have a circular chromosome. So each cell, each bacterial cell, okay, has one circular chromosome, which is called a nucleoid. Okay, and they may also contain small pieces of DNA organized in a circular fashion known as plasmids. Okay, they may have plasmids, and they also have their own ribosomes. Okay, and of course, bacterial cells have the components of all cells, including the genetic material, which you can see there. Okay, also includes the cytoplasm, or the liquid inside of the cell with the ribosomes in it, and they also have membranes that surround them. Okay. So those are three components of any cell. You have genetic material, you have a cytoplasm, and you have membranes, okay? Now there are three different types of genetic recombination in bacteria, okay? One of the types are called conjugation, all right? So conjugation sort of refers to DNA being transferred from a donor cell to a recipient cell through a structure known as a pilus, okay? So that's illustrated here. Okay, so the so-called one that's designated in quotation marks male is the one with the pilus that you see here, okay? And the recipient cell, the recipient cell is the one that's designated as the so-called female cell, and that's the one that receives the genetic material from the so-called male cell. You see it in here, okay, the pilus being connected in the second illustration from the top. And then you see here the genetic material being transferred from the so-called male cell into the so-called female cell here. And then you have the old donor cell, then the new donor cell. Okay, so that's one type of genetic recombination called conjugation. So that conjugation refers to like conjugation as in conjugal rights having to do with a male and a female, you know, the marital rights between a man and a woman who are married, okay? Now the second way that bacterial cells do genetic recombination is called transformation, okay? Transformation is when the bacterial cell just happens to be in a particular environment, and the cell gets pieces of DNA that has been released by cells that are already dead. So the bacterial cell just kind of picks up DNA from the environment it's in. Okay, so here we have a bacterial cell here, which is oval in shape and brown. And then under that you see there a rectangular bacterial cell. Okay, it's just designated rectangular. And you can see the genetic material there in yellow being taken up by the rectangular cell. And then that piece of DNA is then in this recipient cell being incorporated into the plasmid there, okay? So that can happen. Okay, that's called transduction, or I'm sorry, transformation. And then thirdly, you have transduction. Okay, transduction happens when a virus of some kind, okay, a virus or a vector, or viruses acting as vectors, carry DNA from one cell to another. That's illustrated here. There you have a bacterial cell with a circular chromosome, and on top of it you see a virus. Okay, the virus will have injected its DNA into the bacterial cell. Okay, and then that actually takes over the bacterial cell, programs the cell to actually make more virus particles, and the virus particles come together, okay, and then lyse the cell. Okay, then those viruses then infect another bacterial cell or more bacterial cells and inject its DNA into the bacterial cell, and then the DNA is incorporated into the nucleus of that other cell. Okay? So we have conjugation, we have transformation, we have transduction. Okay? Now there are three main forms of bacterial cells in terms of their shape. You have the cocci type, singular coccus, okay? And these generally are spherical kind of singular cells, spherical cells, all right? The second main type of form that bacteria come in are called bacilli, singular bacillus, okay? And these are rod-shaped, all right? And the third type is called the spirilla, singular spirillum, and these tend to be spiral-shaped or comma-shaped, okay? Now, bacteria are also distinguished by Gram stain, okay? In 1884, somebody named Christian Gram discovered that bacteria could be grouped based on their cell walls' reaction to dyes, okay? So first of all, they would heat fix a smear stain with violet dye, okay? Then they would add either dilute alcohol or iodine or acetone, okay? So some species will lose the violet color, but then they'll absorb the pink safranin. So these kind that absorb the pink safranin and lose violet color would be called gram-negative bacteria, okay? Named after Gram, okay? And the gram-positive bacteria will retain the violet color, okay? So here we have an image. You have the pink gram-negative bacteria, seen here as rod-shaped bacteria. And then here you have the violet-stained gram-positive bacteria, okay, with the violet coloration, the more rounder shapes. Okay. Next, we're going to talk about the ways that they can be used, bacteria. Okay, so one way bacteria are used are for bioremediation. Okay, and the definition of bioremediation is the use of living things to clean up either toxic wastes or pollution. Okay, so what they have found out is that bacteria can break down certain kinds of compounds, especially some toxic compounds such as nitroglycerin and trinitrotoluene. Okay, trinitrotoluene, I'm sorry. Okay, and so they could use these bacteria to kind of detox areas around munitions factories and explosion sites. Okay. So Pseudomonas cerevisiae can clean up oil spills, okay, and also chemical dumps containing degreasers. Okay, that's a image of this particular bacteria, Pseudomonas. Okay, they include these different compounds: TCE, creosote, which you find in telephone poles. You know, that smelly stuff that they put in telephone poles to keep them from being eaten by termites, and also 2,4,5-T, which is a insecticide. Okay. Some people believe also that bacteria may in future be used to break down nuclear waste. That's a big exclamation point there. Okay. Now, bacteria can also produce products that's important in industry, such as curing vanilla pods and cocoa beans, okay, coffee, okay, black tea, sauerkraut, dill pickles, vinegar, because these are all products that are important foods, okay, and food products that we know of that bacteria can produce, or that we use to produce these different products. We use bacteria to produce these different products. Okay. Now viruses. Okay, so we're finished with bacteria for now. Okay, so we're going to go into the viruses. Remember that viruses are not really considered to be alive, right? And viruses are actually pieces of DNA or RNA, and they're surrounded by a protein coat, okay? And viruses are responsible for certain infectious diseases such as smallpox, okay? So there's an image of the smallpox virus, which is, of course, killed probably millions of people over the hundreds of thousands of years, okay? So smallpox has been widespread for thousands of years, killed lots of people. We can't even think about how many people smallpox has killed, okay? And it causes these blister-like lesions, okay, which lead to the formation of pits on the skin if the person survives, okay? Here's a photograph of a man that's been infected with smallpox. You can see all the deforming sort of sores on his face. And in 1901 in New York City, or in New York, 720 died, okay? And supposedly smallpox has been eliminated in the United States by vaccinations. Okay, I myself was vaccinated with a smallpox vaccine. Almost everybody I know has been, but I think they might have discontinued that, I think, in younger people now. But what they do is they introduce a weakened form of the virus into the person, and this antigen produces antibodies by the person, and that actually leads to the person becoming immune to the smallpox virus. Okay. So smallpox, they knew, was caused by something smaller than bacteria. Okay. And these agents of disease, this is like historical, they could pass through filters, or what they call filterable viruses. Okay. So some of the kinds of diseases that viruses can cause include some really nasty things, such as smallpox, like we were just talking about, rabies, okay, rabies and measles and mumps, okay, chickenpox, polio, yellow fever, the flu, fever blisters, like you see in this person's lip, warts, and the common cold. Okay, so you have all these nice images here, all these nice small images that we can see of different kinds of viruses and some of their effects. Okay. Now, viruses do not divide. Okay, they do not grow or increase in size. Okay, they do not respond to external stimuli. Okay, they cannot move on their own. They have no independent metabolism. Okay, but once inside their host cell, inside a living cell, they can express their genes and produce more virus particles, like we saw when we were looking at the bacteria. Okay. Now about viroids and prions. Okay. There are more than a dozen plant diseases called by viroids. Okay, so for example, chrysanthemum stunt disease. Okay. And also potato spindle tuber disease. So on the right there you see a healthy potato, and on the left there you see potatoes that have been infected with these viroids that cause a spindle tuber disease. Okay. So what are viroids? Okay. They're circular strands of RNA that live in the nuclei of plant cells that have been infected with them. Okay. And they're transmitted from one plant to another by pollen or by their ovules or the machinery that actually goes along with pollination. Okay. Along with machinery, you know, that is used to harvest plants and etc. Okay. Now, remember, so viroids cause plant diseases, and prions are actually agents that cause diseases of animals. Okay. They're particles of proteins that cause diseases of animals, right? And we've heard of mad cow disease. Okay, we heard of scrapie. Okay. Also, Creutzfeldt-Jacobs disease and chronic wasting disease are caused by these prions. Okay, and these are diseases of humans. They induce the abnormal folding of proteins in the brain. Okay, and that can, of course, lead to brain damage. Okay. Whenever you have anything going on with your neurological system, you can have damage to the nervous system, and this particular disease is always fatal, always ends in the death of the person. Okay? Okay, now the thing is, life. All living things actually began in oceans. Okay? Less than 1 billion years ago, okay, all living things were in oceans. Okay? And in the oceans, of course, they were protected from drying out. They were also protected from UV light, UV ultraviolet light or ultraviolet radiation. And of course, because water is a moderator of temperature, organisms were protected from large fluctuations in temperature. Okay? They could also absorb the nutrients from water. Okay? But the green algae, approximately 400 million years ago, made the transition from water to land, okay, and became what we know of as green plants, okay? So an example of that, they think maybe some kind of alga related to Coleochaeta, okay, which is a tiny freshwater green alga, actually made the big step from water onto land. Okay. Now, algae themselves, okay, some of their characteristics. They have no true leaves or flowers. Okay, they just have a body known as a thallus, which is like, they're not differentiated into tissues. Okay. So algae include seaweeds and pond scums, fish tank films, and the colored patches in swimming pools, as you can kind of see if the swimming pool doesn't have enough chlorine in it. Okay. And they're grouped into phyla or divisions based on the forms of the reproductive cells and the combinations of pigments, okay, that they have, and food reserves that they store. Okay. For the phylum Chlorophyta, or division Chlorophyta, these are the green algae. Okay. So there are some of them that are unicellular and microscopic. Okay, one example of that is Micromonas, which is only 1 micrometer in diameter. That's like a millionth of a meter. Okay. And this particular alga is the smallest eukaryotic cell that's known. Okay, that's just what we know about. Okay, and that doesn't count all the unknown things that maybe people haven't seen or recorded yet. Now, others in the phylum Chlorophyta are larger. So here's a nice little poster here of different kinds of chlorophytes or green algae. Okay, so they form thread-like filaments in some cases. Okay. Or plate-like colonies of individual cells, okay, net-like tubes, okay, hollow spheres, okay, and some are seaweeds, and they can look like lettuce leaves or long green ropes, Okay, so those are the chlorophyta. Okay. Okay, next, we're going to go into the phylum Chromophyta. Chromophyta, okay? And these include the yellow-green algae, illustrated here, the golden-brown algae. Remember that algae are grouped according to pigments, too, that are present in them. The diatoms, single-celled algae, and the brown algae, which some of them can be huge, okay? So for the phylum Chromophyta, about 3/4 of 7,600 species are microscopic, and the features that they share is that their food reserves are the same, either oils or xanthins, okay, fucoxanthins. They have specialized pigments, fucoxanthins, okay, and they can be unicellular, like the diatoms. So the diatoms are unicellular. Okay, here's a nice picture of a diatom. Okay, you can see the transparent cell wall. Okay, usually the colder the water, the greater number of diatoms that are present. Okay, so in warm tropic seas, they're not as numerous as in the Antarctic and in the Arctic, okay, where they form very large populations. Okay. And a lot of the foam at the wave line on beaches are from the oils of diatoms, because diatoms store oil. Okay. Also, diatoms are dominant on damp cliffs, on tree bark, in bare soil, and on the sides of buildings. They're like tiny glass boxes with lids. They're single-celled algae. Okay. So about half of the cell wall will fit into the other half, okay? So many marine diatoms are circular in their shape. They're like tiny little circular glass boxes, okay? So lots of freshwater species resemble a kayak, okay? So up to 95% of the wall is silica, and that's like glass, yeah? They're deposited in pectins and other biological compounds, okay? And the walls of the diatoms have these grooves and pores, and the diatom walls also connect the protoplasm with their environment, okay? Okay, those are the diatoms. Now the brown algae are the Phaeophyceae. They used to actually belong to their own division, but now they put them only in a family, okay? And these are macroalgae. They're brown to olive green, okay, due to the pigments present in them. They, like I said, were in their own division or phylum before, but they do have close relationships with others of the Chromophyta, including the diatoms, okay? Many of them are very large. Okay, none of the brown algae are unicellular or colonial. They're all macroalgae. Okay, there are only a few in fresh water. Okay, and most of them are in the colder ocean waters, open ocean, or some places where like they form the Sargasso Sea. Okay, so Macrocystis perifera, okay, is a giant kelp. Right? It's actually can grow in 100 or more feet, okay, in deep water. And there was actually one that measured 900 feet tall, amazingly. Okay. Larger than any tree, I think of. Okay. So they have a multicellular body known as a thallus, and usually it's flattened. Okay, there's actually looking like a, and they're not organized into leaves, stems, and roots. They're not organized into organ systems. Okay. So they have a holdfast, stipe, blades. Okay, these are parts of them, but they're not really organs per se. So blades and most of the body can do photosynthesis, and many of them have these bladders that help keep them buoyant. Okay, their colors are light yellow brown to almost black. So you have these pigments, fucoxanthin, you have chlorophyll A, okay. I think chlorophyll B, chlorophyll C. Anyway, here's a look at a brown alga, okay. You can see the bladders, you can see the blade, okay, the stipe and the holdfast. Okay. Now one that we're familiar with here in Hawaii is sargassum. Okay, I think it's Sargassum echinocarpum. And the Sargasso Sea in the Caribbean gets its name from floating seaweeds. Okay. So Sargassum echinocarpum, okay. Chopped up was used by Hawaiians as a poultice for cuts from coral. Okay. And the limu kala was used for ho'oponopono, which is like a healing ceremony, okay, for people when there was a disagreement in the family, okay, or in the community. They would have a healing. People would talk out their wounds and frustrations, and then they would eat a little piece of this limu kala. Okay. Okay, now the rhodophyta, or also known as the red algae. Okay. Now most of them are seaweeds in warmer areas and in brown algae, okay, and also deeper. Generally, the algae that are closest to the surface of the water tend to be the green algae. The next deepest would be the brown algae, and the deepest would be the red algae. Has to do with the pigments and the refraction, the absorptions of light waves that actually can reach them through the water. Okay. There are a few that are unicellular. Okay, most of them are filamentous. And the filaments are so tightly packed so they look like blades, but they're still filamentous. Okay. They have very complicated life cycles. Okay. And sometimes they can have 3 different structures of their thalli, or their thalluses. Okay. So meiosis occurs on a plant known as a tetrasporophyte. Okay, so here's a look at a life cycle of a red alga. There's a tetrasporangia there in the upper left-hand corner, producing tetraspores. Okay, and you have the growth of the gametophyte, the male gametophyte or the female gametophyte. Okay. The male gametophyte making spermatangia, the female gametophyte with a carpogonial branch. Okay. Then when sperm and egg come together in fusion, that produces a carposporophyte. Okay. Where so you have all these different kinds of bodies that the red algae have, thalli. So the gametes are produced on separate male and female thalli. Okay. And the reproductive cells are non-motile. That means the sperms are not flagellated. Okay. But the zygotes may move from one cell to another through special tubes. Okay. We see here. Okay. Okay, now most red algae are red-purple in color due to the presence of red and blue phycobilins. Remember, phycobilin pigments occur in algae. Okay, I think they also occur in the cyanobacteria. Okay, as in the cyanobacteria that you see here. Okay, so the red algae may be derived from the cyanobacteria. Okay, and they have chlorophyll A, sometimes chlorophyll D in chloroplasts. And their food reserves are include Floridian starch and mannitol. Okay, and some red algae produce agar. Okay, agar is very important in terms of tissue culture. Remember we talked about that before, that we talked about we use these agar to make the media in flasks and in test tubes when you're doing tissue culture, agar, right? So agar is generally from the red algae. Go into that a little later. Now the edible limu, the greens, include the Enteromorpha species, also known as limu ele. Okay. Also ova fasciata, also known as limu parlaha. You need to know the names of these limu. Okay. Also we have Codium eduli, which is limu vaiole, or rat's foot. Okay. And some edible red algae, okay, include Halaminia formosa, limu lepa ahina, okay. Also a favorite, Asparagopsis taxiformis, also known as limu kohu, okay. And then we have Gracilaria coronapifolia, which is known as limu manaea, okay. So you need to know the names of the edible limu. Okay. Now the phylum Dinophyta. Okay. They include the dinoflagellates. Okay. Which can be responsible for sudden red tides. Okay. Which is a big multiplication of dinoflagellates. You just kind of reproduce really fast. Okay. A characteristic of dinoflagellates is they have two flagella in grooves. Okay. One of them trails behind the cell, acting like a rudder, and the other one encircles the cell in the middle, okay? And it creates a spinning motion, okay? So for dinoflagellates, most have two or more chloroplasts, okay? You have xanthophyll, chlorophyll A, chlorophyll C2. About 45% are non-photosynthetic though. Okay, some will actually ingest food. Okay, so here we have the diagram of a dinoflagellate, and so you can see the flagellum around its center. It goes around the center, and then you have one that kind of trails behind it, like a flagellum. Okay. Okay, now we're going to return to the topic of agar. Okay, most agar comes from the Gelidium group, Gelidium genus. Okay, and this agar is widely used in labs and in medical places like hospitals. Okay, it's used as a solidifier of media for bacteria. Okay, so when you have agar and you add nutrients to it, you can grow animal and plant cells in it, okay? So you have pollen grains full-size plants, okay, you can grow. Also, it's used in orchid tissue cultures, okay? Furthermore, Gelidium is also used for capsules for drugs and supplements, okay? It's an additive in baked goods, for, I guess, a texture to make it kind of creamy and smooth, or as an emulsifier. And it's also used as a base for cosmetics. Okay, so we're done with the protistans and the bacteria. Okay, so we're doing a survey of all these organisms that we're studying, yeah, that botanists study. So the next group are going to be the fungi that we're going to look at. Okay. There are approximately 100,000 known species of fungi, and there are more than 1,000 new species described every year. Okay, there could be more than 1.5 million not yet described. Okay, so what fungi are made up of? Hyphae. Hyphae are masses of fungal threads, singular hypha, Hypha. Okay, so this mass of fungal threads can actually form a mass of hyphae known as a mycelium. Okay, and they can grow very quickly, not overnight or even just a few hours. Okay. Of course, fungi are very important to recycling of dead organic matter. Okay, but also because of the way they feed, okay, they cause huge losses through spoilage of food and through disease. Okay, look at that. Wow, you actually see a deterioration of a peach, it looks like. My goodness. Okay, so mycologists are the people who study fungi. Okay, so distinctions from Protista. Okay, so before, the fungi and slime modes and bacteria were put in one phylum of kingdom Plantae. This was before. Okay, but that was before they knew about the difference between prokaryotic cells and eukaryotic cells. So that didn't come along until like the 1800s, okay, a couple of centuries ago. Okay, and then what they did is they put the bacteria in the kingdom Monera. Okay, put it separate from the others. Okay, and so the metabolism and reproduction and lines of diversity were different from plants. So they put the fungi in the kingdom fungi. Okay, so true fungi are filamentous or unicellular heterotrophs. Heterotrophs, remember, means that they need to actually get their food or their energy requirements met through another source. So most of them absorb their food in solution. So they will secrete a digestive enzyme and then absorb the digested food outside of their bodies into their bodies. So some of them live on dead organic material, and they're known as saprobes. And some of them live on living hosts, and these are parasites. Okay. Now some have mutualistic relationships with plants. We've talked about this before for sure. And they're known as mycorrhizae. Remember, mycorrhizae are symbiotic fungi that live in relationships with plants, and almost all plants have mycorrhizae. They're associated mycorrhizae. Okay? So the fungi are in 5 phyla. Okay? They're mostly filamentous. Okay? Most of them do not have cells that are motile or that move. Okay? So the filamentous fungi, they grow at the tip of their hyphae. Okay? And a mushroom actually comes about from the hyphae that are interwoven and packed together. Okay, so if you were actually to get a mushroom and dissect it, you would see that there are actually all these fungal threads in there. Okay. So the cell walls of fungi are mostly of chitin, which is a similar material to what makes the exoskeleton in arthropods. Okay. And these exhibit varieties of sexual reproduction. Okay. They secrete digestive enzymes and absorb food in solution, like we talked about a little earlier. So the first group we're going to look at are the zygomycetes or the zygomycota. And these are the cenocytic true fungi. What does cenocytic mean? Cenocytic means that they don't have cross walls. Okay. It means that they have multinucleate bodies. Okay. So the well-known black bread mold or Rhizopus stolonifer, okay, is among these zygomycetes, right? And of course, their spores are everywhere, okay? In the air above the North Pole. This is amazing. They've been detected over jungles, okay? They've been found inside and outside of buildings, okay? Also in soils, clothing, cars, and all the way far out to sea. So they're ubiquitous, they're everywhere, okay? So those are the zygomycetes. Okay. Now the ascomycetes or the ascomycota are the sac fungi. Okay. So one example of a sac fungus are the truffles. So the truffles, they actually have their fruiting body growing 1 to 6 inch deep in the ground. Okay. And they're found usually near oak trees, and they sort of look like a big prune. but they're supposed to be delicious. I myself have never eaten truffles yet. I think I will try them someday, but they're so expensive. You know, even if you have oil that's been soaked in with truffle, it's expensive, like $20 a bottle or something. Okay. Now, so they can be 4 inches in diameter, so they're kind of large, very large, and you can see they're sort of, they can range from a kind of beigey color to brown to black. Supposed to be really delicious though. Now we're going to talk about ergot rye. Okay, so it's actually a ascomycete that infects rye plants as well as other plants, and what they call ergotism affects people's central nervous system or the central nervous systems of mammals. Okay, it can cause hysteria. Okay, convulsions. Okay, death. Okay, there's another form that can cause gangrene, right? So what they used to call St. Anthony's fire, a symptom of ergot poisoning, ergotism, was common in Europe during the Middle Ages. In the year 994, it killed 40,000 people. Okay. In 1722, the cavalry of Peter the Great was overcome by this disease. Just as he was about to conquer Turkey, they probably ate bread that had been contaminated with ergot rye. Okay, made rye bread of some kind. Okay. So ergot rye is implicated in social upheavals such as the French Revolution. You know, the hysteria that sort of reigned during those days, okay, might have been from this ergot rye. Okay. Also, the Salem witchcraft trials might also have its foundations in ergot rye. Okay. But it also may have medicinal uses, such as it can be used. Okay, here's actually a rye plant or rye fruits with ergot on them. You see those kind of brownish spots on them. Okay. They can initiate childbirth. I guess they stimulate contractions of the uterus. And also, of course, if they can stimulate contractions of the uterus, it can also induce abortion. Okay. It can be used to treat migraine. And it was an initial source of LSD. What do you know? You learn something new every day. Now, the phylum Basidiomycota, okay, also known as club fungi, right? So they number about 25,000 different species that have been named, and they include the stinkhorns, the mushrooms, the toadstools, shelf or bracket fungi, rusts, smuts, jelly fungi, and bird's nest fungi. Okay? So those are different types of basidiomycetes. Okay? Now, in terms of the human and ecological relevance of fungi, there's a Teonanacatl, which translates to God's flesh, okay, which is a sacred mushroom, psilocybe, cyanescens, okay. It was used in religion, in native religions in the United States or in Mexico too, okay. So small amounts of these mushrooms would induce very focused and vividly colored visions. Okay. Another fungus used for similar purposes was fly agaric in Russia and India. Okay, and the use of this fly agaric also dates back many centuries BC. And the God's flesh mushrooms are still currently used by people who are practitioners of Native American religions. Okay, so it is still used in religion today in some parts. Okay, some now diseases that are caused by fungi. There's aspergilloses that infect the respiratory tract. Okay, so there's a fungus called Aspergillus flavus that secretes a toxin known as aflatoxin. This is kind of important. It's actually the most potent natural carcinogen that's known. Okay, it's known to cause liver cancer. Okay, places where there's humid climates. Okay, it can infect peanuts and peanut-based products such as peanut butter and peanut-based dairy feeds. That actually one of the reasons why I stopped eating peanuts, because peanuts are cheap, they're a source of protein, but the aflatoxins can accumulate in your tissues over time. Okay, and so it's good that you know about aflatoxins. Okay. Now lichens. Okay, lichens are a symbiosis between a fungus and an alga, or a fungus and a cyanobacterium. Okay, so the thallus of lichens can be from 0.04 inches to 6.5 feet. Okay, so generally the green alga or the cyanobacterium will supply food. Okay, and then the fungus will actually protect the rest of the plant from light. Okay, and increase rates of photosynthesis. Okay, and absorbs water and minerals, right? So lichens number about 14,500 species, with approximately 20 species using club fungi. Okay, one species using a bacterium, which is of course the cyanobacterium, and others are all with sac fungi. Okay, and about 100 species of algae or cyanobacteria. Okay, and they're generally identified according to fungus. Okay, and they grow very, very slowly. Okay, there are some lichens that actually can be only a couple of inches wide, and they're hundreds of years old. Okay, so they grow slowly. Usually at most they grow maybe 0.4 inches a year, less than half an inch a year. Okay, but they can live maybe more than 4,500 years. Okay, they can also tolerate a wide range of conditions depending on the lichen. Okay, even though they are used in terms of being pollution indices. Okay, so the thallus tends to be 3 to 4 layers of cells or hyphae. You have the upper cortex, the algal layer, the medulla, the lower cortex, and they're grouped into 3 growth forms. So this is basically the layering of a lichen thallus. Okay, upper cortex, algal layer, medulla, and lower cortex. So they're grouped into 3 types of growth forms. Okay, the first type is crustose. Okay, crustose lichens are like crusts. Okay, they're attached or embedded in the substrate over the lower surface. Okay, that's crustose lichens. The foliose lichens tend to be kind of leaf-like. Okay, they can overlap. Okay, and they're not so strongly attached to the substrate as the crustose lichens are. Okay, so we have the crustose lichens, the foliose lichens, and we have the fruticose lichens. Okay, they're like little shrubs, or they hang down. Okay, the thalli are cylindrical and attached at one point. Okay, so now we're entering the kingdom Plantae, leaving the kingdom Fungi, and entering the kingdom Plantae. Okay, so the kingdom Plantae, remember, they're actually descended from green algae, like the Colochaeta, right? So on land, they're kept from drying out. Okay, with their plant surfaces coated with cuticle. Remember that waxy substance made of cutin. Okay. You also have gametangia, special structures to actually make and protect the gametes. And also sporangia, special structures made to produce and protect the spores. Okay. They're multicellular with jackets. Okay. And the plant zygotes develop into embryos. Okay. Now the bryophytes, okay, are the first group we're going to cover. Okay, these are the non-vascular plants. Okay, and they're in 3 different divisions or phyla. The phylum Hepatocophyta, or the liverworts. Okay, the phylum Anthocerophyta, or the hornworts. Okay, that you can see there, little horn there, which is a sporophyte. And then we have the phylum Bryophyta, also known as the mosses. Okay, let's see. Now, of the different kinds of bryophytes, the most important to humans are the peat mosses. Okay, why is that? Okay, so about 2.2 pounds of dry peat moss, okay, can actually absorb up to 55 pounds of water. Okay, makes it very valuable as a soil conditioner and part of potting media. Okay, they also ship live shellfish in peat. Okay, it also has natural acidity. Okay, and if it's acid conditions, it inhibits fungal growth and bacterial growth. Okay, it also has antiseptic properties. Okay, and during the Crimean War and during World War I, it was used as a poultice material for wounds. Okay, 1854 to 1856, Crimean War. Okay, so peat, soil conditioner, can be used for fuel as well. Okay, it's also used to kind of season Scotch whiskey. They're kind of the malt or barley is kind of held over a smoky peat fire to lend the barley the taste of Scotch whiskey. So the sprouted barley is dried over a peat fire, and that gives a smoky flavor to it. So people who love Scotch really love their Scotch. I had a friend who used to drink Scotch on the rocks. Okay, now ferns and their allies. Okay, so these are plants that have conducting tissues. Here's a look at celery, which is a flowering plant. It's not actually a fern or relative, but you can actually see the vascular bundles. Okay, they do not produce seeds. So ferns and their relatives reproduce through spores. Okay, not through seeds. Okay, now in terms of the evolution of vascular plants, so here's a bunch of tall trees. So then in the vascular plant evolution, xylem and phloem develop. Xylem moving water in an upwards direction, and phloem moving sugar or food in any direction from where it's made or stored to where it's either stored or used. Okay, from a source to a sink. Okay. Now, in terms of ferns and their relatives, the gametophyte got smaller and more dependent on the sporophyte. Okay. The sporophyte got larger. Here you see some heart-shaped gametophytes of ferns. Okay. Protected the gametophyte. There's a little embryonic plant there in that structure. So there are 4 phyla of seedless vascular plants, including the Xylophyta, which are the whisk ferns. Okay, the Lycophyta, which are the club mosses and quillworts. Remember, wort means plant. The Ecocetophyta, also known as the horsetails and scouring rushes. And the Polypodiophyta, also known as the ferns. Okay, now we come to the seeds. Okay. So the oldest seeds date to the Devonian period, more than 350 million years ago. Okay. They have protective seed coats and food, and the embryo may lie dormant for a while, so the seed actually helps to protect the embryo and may help the embryo survive freezing or drought or fire. Okay, So seeds do have survival value. So right now on this planet, seed plants are dominant, whether they're gymnosperms or angiosperms. So gymnosperms, of course, are the conifers and others, which we'll go into a little bit later. And then you have the angiosperms of the flowering plants. Okay. So the first seed plants were fern-like. Okay. They left fossils with obvious seeds. So they used to call them pteridosperms. Okay, but now they're known as early gymnosperms. They're called seed ferns, but of course ferns do not have seeds, even though they did sort of resemble ferns. Okay, so you can see here they did resemble ferns, some fossils, right? So gymnosperms, the definition of the word, gymnos means naked, and sperma means seed. So gymnosperms bear naked seeds, okay, or seeds that are exposed to the elements. Now the sporophytes of gymnosperms are mostly trees and shrubs, a few vines, but not many that much to speak of, okay. So the gametophyte develops within the sporophyte, and the gametophyte is dependent on the sporophyte, okay. Now in terms of the phylum Pinophyta, also known as the conifers, okay, there are approximately 600 species. Okay, they include the pines, the firs, the spruces, the hemlocks, the cedars, the redwoods, and etc. Okay. So here's a look at a bristlecone pine, which includes it's the oldest living organisms. Okay, they're more than 4,500 years old, some of them. Okay, they live in the White Mountains and the Snake Range. Okay. Now the conifers have needle-like leaves, and these leaves are arranged in fascicles, usually fascicles of 2 to 5 leaves each. Okay. So fascicles are short shoots with restricted growth. Here's another look at some Pinophyta. Okay, so this is the grove in which Prometheus is in Wheeler Park. Okay. Now these plants oftentimes live where the elements are kind of harsh, where the topsoil is frozen. Okay. So for many of these plants, it will be difficult for the root to get water if it weren't so tough. Okay. Furthermore, their leaves tend to be exposed to high winds and extremely cold temperatures, so these are very tough plants. Okay. So here's a look at these fascicles here with the needle-like leaves. Okay. And there you can see snow on the leaves of this pine. Okay. So that's a phylum Pinophyta. Now the phylum Ginkgophyta includes one particular species called Ginkgo biloba. Okay, so there's only one living species, Ginkgo biloba. Okay, and the word Ginkgo, the Chinese word for it meant silver apricot, and there's actually a story about the name of this plant. That the botanist that went to Japan and saw a ginkgo plant actually wrote down ginkgo instead of ginkgo. Ginkgo means silver apricot or silver nut, you know. So it actually was a mistake that he made. He wrote down ginkgo instead, but now it's known as ginkgo from a mistake. Okay. Now fossils show that the Ginkgoaceae, this family, were once widely distributed in the northern hemisphere, and until they discovered it, Westerners discovered it in Japan and in China, they thought it was only known through fossils. Okay, so they call it a living fossil because for many generations, or probably hundreds of years, they thought it was known only in its fossil form. Okay, so here's a look at some ginkgo leaves. Okay, take a look at the leaves, And they're often called maidenhair trees because their fan-shaped leaves kind of look like the leaves of maidenhair ferns. They're widely cultivated in the mainland United States. They are widely cultivated in the mainland United States. Okay, leaves don't have a midrib or prominent veins, and you have these hair-like veins that branch dichotomously. Okay, here you see a female structure from a female plant. Okay, and male structure from a male plant on the left. Female structure is on a female plant on the right. Okay, so those will develop into the seeds. Okay, so here's some seeds to remind you that these gymnosperms and the angiosperms are seed-bearing plants. All the other plants that we talked about before, the fern allies and the bryophytes, they all bear spores. Okay. Now another phylum is the phylum Cycadophyta, which are known as the cycads. Okay. They sort of look like, between a tree fern and a palm, but they're not related to either tree ferns or palms. Okay, they're in their own group. Okay, they're a gymnosperm. They are slow-growing plants of the tropics and subtropics. They tend to be toward more warmer places. Okay, they have unbranched trunks. Okay, some of them growing over 50 feet tall. Okay, they have a crown of large pinnately compound leaves, as you see in this particular individual. Okay, so there are approximately 300 species, and there are several of them that are facing extinction. Okay, because of overcollecting and also habitat destruction. Okay, so their life cycle is similar to that of conifers, and their pollination are sometimes affected by beetles. Now this is an interesting factoid. Each sperm has 10,000 to 20,000 flagella. Okay, it's kind of interesting factoid there. Now they're dioecious. Do you remember what dioecious means? Okay, dioecious means you have two houses. You have a male house with the male structures, a female house with female structures, right? So those are when you have two plants, you need a male plant and a female plant to actually affect sexual reproduction. Whereas monoecious means that you have male structures and female structures in the same plant, like male flowers and female flowers, or male cones and female cones, as you do in the conifers, okay, in some cases. Okay, so you have the pollen structure and the seed structure are on different plants. Okay, and in some species the strobili are huge. It can be more than 3 feet long and can weigh over 100 pounds. Okay, so this is looking at a male structure here that produces a pollen. Okay, now the phylum Cnidophyta includes the cnidophytes. Okay, so these include 70 species in 3 different genera. They're not that closely related to each other. The Cnidophyta are like a catch-all phylum, all right? That they'll put some that they all actually have certain characteristics, like they all have vessels in the xylem, but otherwise they're not that closely related. Okay, so they have vessels in the xylem. So about half of them are ephedra species, which are also known as the joint firs. Okay, these are shrubby plants in southwestern North America in the dry areas. They have tiny leaves, okay, 2s and 3s at nodes, and they tend to turn brown. Okay, this is sort of what it looks like. Okay. Okay, their stems and branches are slightly ribbed, okay, and photosynthetic when they're young. Okay, we also have gnetum. This is gnetum, Gnetum, gnetum niemann. Okay, and there are the developing seeds there on that structure. Did you see it toward the middle of that photograph. Okay, so most of the other species in this phylum are the Needham genus. Okay, so these are plants that live in the tropics of South America, in Africa, and in Southeast Asia. Okay, you have most vine-like with broad leaves, but the best known is a tree that grows up to 30 feet tall. So you have Needham, Neiman in Foster Botanical Garden, in their prehistoric garden there downtown, across from Zippy's. Okay, here's some male structures in Needham. Okay, now the third group is Welwitschia, which is just one species. It's a monotypic genus. Okay, so it lives in the Namib and Mossamedes deserts in southwest Africa, where annual rainfall is 1 inch or less, amazingly so. So they do cam photosynthesis, right, where the stomates are closed in the daytime and the stomates are open at night. Okay, so the stem of this plant only rises a little from the ground. That kind of helps protect it from the high winds. And the stem is in the form of a cup, which tapers into a very long taproot. Okay. They may live to be 100 years old. Okay. And stems may be more than 3 feet wide. Okay. And they produce only 2 leaves. Okay. And these leaves are wide in terms of their physicality, and they have a meristem at the base. Okay, and these plants are also dioecious. Remember, male in male plant, female in female plant. Okay, so here's a look at Welwitschia. Okay, so that's a plant, a very unusual plant that I actually saw at Huntington Garden years ago. Okay, very interesting plant. Okay, so what we'll do is we'll stop here for the first part of our review session, and we'll have a second part that will be coming up soon. Okay, so you can watch it at your leisure. Okay, see you next time. Bye-bye.